All right, so this is video 1 of 2 for your section 2 notes for chapter 5 in biology. In this section, we're going to talk about the structure of a chromosome, and we're also going to follow those chromosomes through the process of mitosis and cytokinesis. We're going to cover the first objective in the first video here, and then talk about mitosis in another. So first definition, you have a chromosome, right? Chromosome is a continuous thread of DNA containing numerous genes. So this is where all of your genetic information is stored, right? So DNA, you already knew, held your genetic information, but what you may not have known is that you have many pieces of DNA, right? You have 46 different chromosomes as a human. Other organisms have different numbers of chromosomes, but that chromosome is just one continuous thread of DNA, right? And you have many of those. Histones are proteins that your DNA is wrapped around, right? You need to protect your DNA, obviously, so a lot of times you need that DNA wrapped around these histones and actually coiled up fairly tightly to keep it from being damaged. We're actually going to talk about two different forms of DNA, two different conformations that it can take on. Chromatin is a very loose combination of DNA and histones that you're going to see during interphase, right? When chromosomes are in their chromatin form, right, they're actually not visible in a cell. You can't see them. DNA, a piece of DNA, a chromosome, can be really, really long. It can be as long as your arm, but it's so thin that it's invisible, right? So when you don't have them wrapped up very tightly, you can't see them in a cell. And this chromatin form is what you're going to see during interphase, because in interphase, you're actually going to read your DNA. You're actually going to use it, right? Your cells are going to be doing their normal functions. So chromatin is that loose form of DNA, right? It does say that that chromosome is still tightly wrapped around the histones, but the DNA is not going to be supercoiled, right? We're going to talk about that supercoiling later, because I might not be able to describe that very well in a video. But chromatin is the invisible version of your chromosomes, so maybe we could put that in here, right? Chromatin is invisible for all intensive purposes, hard to see. Whereas a chromatid form of your DNA, that's when you actually can see it. This is the condensed version of your chromosomes, and you can see it. It is visible, right? And a chromatid is 1/2 of a tightly wound chromosome, because technically your chromatids, you should have 2 of them per chromosome, because that DNA is going to be replicated before you're going to divide. So during mitosis, you have to start moving your DNA around. It needs to be in a condensed, compact form to be moved around without being damaged. So again, we're going to talk about the supercoiling for that later, but for your purposes, you need to know that in interphase, when you're using your DNA, when you're reading it, you can't see it. During mitosis, when you're moving the DNA around, it's tightly packed. You can't be using it, right? But it's packed up tightly so that it's protected, so you can move it around without ripping it apart. So we're going to talk about different parts of a chromosome, but we're talking about chromatid forms of chromosomes when we talk about that, because we can't see it otherwise. Right? We're going to have a centromere, a central point where sister chromatids are attached. Or held together, I guess, is what it says over there. So there's a little bit more to that than I could see. So if I can insert a page here, right? If I'm talking about a chromosome, typically you're thinking about this. Excuse me, that's kind of ugly. Let's try that again. Typically you're talking about this condensed version and this X shape that we're familiar with. So again, if you can see it at all, it's in chromatid form. And I have 2 chromatids here that are connected at this centromere. right? It's a central point. It's in between, right? And connecting those two chromatids, right? And it's just where they're held together currently before I divide them so that each cell has one copy each. So centromeres, something you need to know. You also need to know what a telomere is, right? And the telomeres are actually at the ends of your chromosomes, right? And there's just repeating nucleotides that are actually nonsense. They don't code for anything. And the reason for them is to protect your chromosomes, right? When it's all said and done, again, we're going to be moving these things around and we don't want them to get damaged. But where the most damage can occur is typically at the ends of these chromosomes, which is why you have those telomeres, right? It's just like the end of a rope that's more likely to fray, right? You have these telomeres to protect the ends of your chromosome just in case, right? So the centromere holds them together. That's just that central point where they're connected. And then the telomeres are on the ends of those chromosomes because that's where the damage is most likely to occur, right? As an added note there, the ends, again, are most vulnerable to damage of any kind, so you have to protect them. But it also says down at the bottom there that you lose a little bit off the end of a chromosome every time you replicate it, right? So when I say that, right, every time I copy DNA, right, these strands get shorter and shorter and shorter. I lose a little bit of DNA every time, right? So it's actually important that those telomeres are nonsense because they're going to disappear. And if they're necessary, that's going to be a problem. And that's actually one of the things that can be connected to this section, right? When we're talking about telomeres and damage and replicating DNA, right? There's a reason why organisms age, right? We don't know all of the complex reasons that are involved with that, but one of the things that's hypothesized is that when you have cells like, say, your skin cells that replicate themselves very frequently, right, as those telomeres continue to shorten, right, as those chromosomes continue to shorten, and you lose a little bit every time, eventually you run out of telomere and you start dipping into actual important stuff that can be damaged. So when I say that a little bit is lost every time, that's actually hypothesized to be one of the reasons why we age, right? Some of those cells that noticeably change when organisms get older, right, like their skin, right, may not be the way it once was, right? It might be wrinkled. It might not be as pristine and perfect as it once was, right? And that may have to do with just damage in those chromosomes as they've been replicated over and over again. So that's one little add-on, right? Maybe not necessarily a test question, but it's just kind of an interesting note. One of the reasons we age is because our cells have replicated so many times that we actually lose DNA, and that damage can add up. So again, just know what a chromosome looks like. You have a centromere holding two chromatids together. You have these telomeres that are important for a couple reasons to protect that chromosome, right? Even from its own replication that causes you to lose a little bit every time. But otherwise, this last note before we move on to the next video, interphase, again, we did a whole circle in our last set of notes that put a bunch of things together so that we could see those different parts of the cell cycle. And we had a big chunk for G1. We had synthesis. We had G2, right? And then we had mitosis itself, right? And we're going to talk about mitosis in this section, right, in the next video. But this portion here is that mitosis. We'll go on to that later. But those three other portions, remember, G1 plus S plus G2, right? All of those things combined are interphase. So when it's all said and done, right, that's when a lot of important things are happening in a cell. That's when the cell is actually doing its normal functions, remember. But in terms of cell cycle, I have to have that growth. I have to have that DNA replication. But once all that stuff's done, and once I pass some checkpoints in G2, I need to divide the nucleus, and that's what mitosis is all about. So our next section of the notes is about mitosis. The only last thing that maybe we need to understand, again, just from what we've been talking about in this section, is if a cell is in interphase, right? If I had a cell over here this time, and I have a nucleus in that cell, in interphase, the DNA is invisible. I told you that I can't even really distinguish between G1S, G2 when I'm looking in a microscope, right? But you'll know when you're in mitosis, because once a cell enters mitosis, that's when you see those condensed forms of your chromosomes. So you'd see at least one, if not many. You'll see 46 chromosomes in a human cell. So chromatin versus chromatid form, right? In interphase, your DNA is invisible. In mitosis, it's actually visible. You can see it. Right, so that should be good for this video.